Thank you, everyone. So I'm really excited about this learning seminar series, and it's very exciting to see our friends from the Fellow Center on the opioids and overdose. So thank you for joining us. We learned a lot the last time we did this, and definitely look forward to hearing what you have to say, Jackie, and everything. So with that, I'm not going to say much other than the format is 5 minutes of telling us something cool that you've read about, 5 minutes of telling us the cool stuff you're doing, and then there'll be a fun Q&A after that for 5-ish or so minutes. And with that, I think I'm just going to introduce our first speaker, which is our very own Ken Wakabayashi, who's in the Department of Psychology. He's a project leader in the RDAR Center, and the title of his project is Psychostimulant Addiction and Nutrient Sensing Neurons, Genetic Targeting to Parse Neural Pathways. It's all yours, Ken. Look forward to it.
All right, thank you everyone for taking some time out to learn something and for me to present both a cool paper and also some of the research that I plan on doing with the COBRE and with the RDAR Center. Today I'm going to be presenting a paper by Noble and Kanonsky, which just published in 2020. It's from Nature Communications, and it was titled Hypothalamus Hippocampus Circuitry Regulates Impulsivity by Melanin Concentrating Hormone. Now, because of the time constraints, this is going to be a highlight summary. It's going to be very broad. Essentially, it's going to be a walking tour. Essentially, when I was a college student, I went to the Louvre for an hour, and I walked straight past the Mona Lisa and said, there it is. So pretty much this summary is going to be that way. I'm trying not to go too much into the weeds or technical detail. But the reason why I wanted to present this paper was because it's very similar in terms of the neurotransmitter system and the circuit that I'm looking at, and also employs a multi-level approach and very similar methods to what I plan on doing with my RDAR project. So in terms of the background and objectives of this paper, broadly speaking, melanin concentrating hormone, or MCH, is a neuropeptide neurotransmitter that's expressed in neurons from both what's called the lateral hypothalamic area, or LAHA, and the zona incerta, which is also now renamed as the incertal hypothalamic area. And the reason why it's interesting to me is that it's associated with food, alcohol, and drug-seeking behaviors. Now, MCH receptors are widely expressed throughout the brain, which means that these neurons interface with many other brain systems, for example, the mesolimbic and the extended amygdala systems that are really important for both alcohol and drug-seeking behaviors. But also MCH-R1 has been found in the ventral hippocampus, or VHP. And the ventral hippocampus regulates both impulsive and motivated responding to palatable food, as well as classical learning and memory. So for neuroscientists, this is the place where LTP was first discovered and highly studied as well. But what's interesting is that the functional significance of these neurons and the receptors in the ventral hippocampus is relatively unknown. So the objective of this study was to determine the role of ventral hippocampal MCH signaling in regulating behavioral impulsivity. Now, in terms of impulsive behavior, one type of impulsive behavior is called impulsive action. And the way that they modeled this in rats was having them being trained to lever press for a high-fat, high-sucrose reward pellet on what's called a differential reinforcement of low-rate schedule, or DRL task, which essentially measures impulsive action because it requires a rat to withhold a lever press for more than 20 seconds. If they lever press more than or less than 20 seconds, these lever presses are not reinforced and they don't get the yummy, fatty, sweet pellet. And so in this study, they activated the MCH system pretty much by two different ways. They injected MCH directly into the brain, and this essentially activates MCH receptors. And they also used a more modern approach, which was to target artificial excitatory designer receptors exclusively activated by designer drugs, known as DREADs, which essentially allows you to specifically target MCH neurons genetically and activate those using a compound called clozapine N-oxide. And what you see from the paper here is neurons in this is the zona incerta, and if you can see the M cherry, which is the virus or the dread, and MCH, which is the protein or the neurotransmitter. So if you overlap the neurons that are expressing both the dread and the neurotransmitter, you can see that there is actually an 84% overlap. So that means 84% of the neurons are transfected with this artificial receptor. And so, and again, I'm just choosing two figures from this paper, but if you look at one measure of impulsive action, which is efficiency, which is calculated by the number of active lever presses versus the number of rewards that they earn, which is essentially a behavioral measure of the impulsive action in this task, it was found that both when you inject and activate the receptors, you decrease efficiency. So they essentially have more impulsive action, and the same thing happens if you activate the neurons themselves. So this paper actually shows that centrally activating either the receptors or the neurons increases impulsive action on this particular impulsivity task. So they also looked specifically at one specific projection. So they looked at the projection going to the ventral hippocampus by using a more complicated, more sophisticated retrograde virus system. And I'm not going to go into the details of how this actually works. Of course, we can talk about it. But essentially, this is a way to express dreads only on neurons projecting specifically to the ventral hippocampus. And both injecting MCH into the ventral hippocampus, activating the receptors, or activating the neurons themselves with CNO reduces efficiency in this task as well. So they were able to conclude that activating this specific pathway also increases impulsive action. So there were some other behavioral tests. I'm going to spend even less time on this, but they were able to show that MCH injected into the ventral hippocampus also increases impulsive choice, which is another kind of impulsive behavior, using what's called a delayed discounting task. Essentially, the rat has to choose between a small reward right away or a large reward where they have to wait a particular delay. And essentially, it shifts the preference towards small immediate rewards. And they did a whole bunch of control experiments, like most behavioral studies, and they showed that this activation has no effects on a lot of different things, such as food eating, motivation, general activity, or time perception. Okay. So one of the things that they also did in this study was they used what's called an audio-radiographic 2-deoxyglucose method, or 2-DG uptake. And essentially this method is a way to indirectly measure brain activity via increased metabolism of glucose via regional cerebral glucose uptake. And why I thought this was interesting, this is the first time I actually saw this technique analyzed in this particular way, but what was interesting was they use this essentially as an analog of fMRI, using a preclinical approach that's using an isotope-tagged compound, which means that there is no magnet, but the animal is not alive. But they were able to show that the MCH injected into the ventral hippocampus actually increased the activation of the nucleus accumbens shell, which is part of the mesolimbic system. So in terms of really broad results and conclusions, they were able to show that targeted activation of the system, both by pharmacological and direct activation by DREADS, increased impulsive behavior, both action and choice, and that targeted activation by DREADS specifically to the ventral hippocampal pathway also increases impulsive action, and that the pharmacological activation also increases impulsive choice. So the overall conclusion is that the MCH system is increasingly viewed as a research target of interest by large, well-funded groups centered around behaviors related to addiction, impulsivity, and obesity. Now, why this is relevant to my COBRE project is that I'm essentially looking at the same neurotransmitter system. I'm using very similar genetic targeting approaches, and I'm also using a multi-level approach, although slightly differently. So much like the study, it's been shown that MCH receptor antagonists, specifically in the mesolimbic area, such as the nucleus accumbens, attenuates cocaine seeking in rats. And it's also been implicated in methamphetamine and alcohol use disorders. Yet little is known about what these neurons do during drug self-administration, specifically that model human methamphetamine and cocaine addiction. So my COBRA project seeks to use custom viral dread vectors that I've developed to look at the contribution of these neurons in rat models of methamphetamine and cocaine addiction. And so you can see my version of the virus that I'm using to express essentially dreads in the lateral hypothalamus. And my strategy is going to be either to activate or inactivate MCH neurons, either in the lateral hypothalamus or the insert hypothalamic area, while the rats are self-administering drugs. Also, we know that experience with cocaine changes how the brain reacts to drugs at a behavioral timescale. So, for example, my previous research has shown that IV cocaine can increase extracellular glucose in the nucleus accumbens. And we also know that MCH neurons activate when brain concentrations of glucose rise because they're directly sensitive to glucose levels. And so one of the other questions that I'm going to look at in my COBRA is to look at whether or not the activity of MCH neurons is correlated with brain glucose levels during IV meth or cocaine. And so the approach that I'm going to use is slightly different from the approach that was gone over by the paper. I'm going to use a combination of enzyme-based glucose biosensors that can measure glucose levels in awake-behaving animals at a second time scale. And I'm also going to be using a custom virally mediated calcium imaging, otherwise known as fiber photometry, to look at the activity of MCH neurons directly in response to IV meth or cocaine. And so, for example, on the right here, I'm showing you a schematic of what calcium imaging sort of looks like. So you have something that is sensitive to an action potential, and it's a protein that fluoresces when an action potential happens. And you are watching this through various different methods, but in this case, we're going to use a fiber optic ferrule or fiber optic cable that's directly implanted into a rat brain. And here I'm showing you my first UNL image, actually, that was captured yesterday, showing viral mediated calcium indicator expression. This is the technical name of it, showing that it's expressed in the rat lateral hypothalamus after fiber photometry. So some other considerations. I also have some ideas about using retrodreads, much like the paper, although that's not currently in the COBRA at the moment. And some interesting things for me to think about as the next steps beyond the COBRA project is looking at what are called long loop connections, essentially also targeting the neurons that are expressing the receptor as well as the neurons that are expressing the MCH neurotransmitter. I'm also interested in sort of branching out to non-drug addiction related behavioral models. And because I sort of ran into this technique with the 2-deoxyglucose, I thought it might be something to think about in terms of interfacing with the human imaging people at RDAR, especially because it doesn't need an animal fMRI or magnet, which is sort of very expensive. So I don't have specific experience in this, but I do have experience in related approaches. So it's just something that I thought maybe would be interesting to look at in the future.
So it's my pleasure to then introduce our second speaker, Tierney Lorenz, who's in the psychology department. She's running a pilot project investigator in the center. And the title of her project is Predictors of Sex-Linked Marijuana and Alcohol Use in Sexual Minority and Heterosexual Women. So take it over, Tierney. It's all yours.
Great. Awesome. Hard act to follow, Ken, but I'll do my very best. So thanks for giving me the opportunity to talk about my project. I'm going to go ahead and pull up my presentation here and try and share my screen. Just a moment. Hopefully you all can see my slides here. Yes? Somebody? Yeah? Okay, thanks. Thanks, Jackie. Jackie's the only one I can see right now. Like Rick mentioned, my project is on sex-linked alcohol marijuana use in heterosexual and sexual minority women. Okay, great. So the paper that I'm going to be presenting on was published in 2019 in the Journal of Sexual Medicine, and it was part of a large series of reports that came off of this massive online survey of drug use amongst English-speaking countries. And so they looked specifically at people who use drugs or alcohol in the context of sexual experiences, what they call substance-linked sex. And in kind of setting up this paper, the authors talked about how the vast majority of the research on the use of drugs and alcohol in sexual contexts has really focused in on men, and in particular on gay and bisexual men's drug use in a very narrow range of contexts, specifically using party drugs like GHB or MDMA in kind of chemsex environments, which is when people come together to use drugs and engage in sexual activity, usually quite a lot of sexual activity, unprotected sexual activity with a variety of different partners in a short period of time. And because of the public health risk that's associated with that kind of sexual behavior, there's been a lot of attention paid to the predictors of that kind of drug use. But the authors of this paper point out that people use drugs and alcohol in sexual contexts in a lot wider range of contexts. And we have very little information on women's use of drugs and alcohol in sexual contexts. We also surprisingly have little data on heterosexual use of drugs and alcohol in sexual contexts. We don't really have a great sense of how unique this is to the sexual minority population. And like I said, we don't have great data on people using drugs outside of these kind of narrow range of situations, these chemsex or situations. So they did this very large scale study looking at broad population level data to look at predictors of people's drug use. And another piece that I really appreciated about this paper is that they pointed out the fact that the majority of research that's been done on women, what little work there has been done, has really kind of taken this narrative that when women use drugs and alcohol in sexual situations, that they're doing so in order to reduce their inhibitions or to reduce their self-consciousness or their sense of shame. But that's kind of only half of the picture, right? You can use drugs to reduce your inhibitions, but you could also use it to enhance your pleasure, to enhance the positive aspects of sexuality. And there just isn't a lot of great data on people's use of drugs for that purpose. And so they set out to correct for that. So they did this large online survey of over 22,000 people. These were all self-report data and they're all self-selecting. So we kind of have to keep that in mind as we interpret the findings. And it was English speaking, or the English survey. And so the majority of respondents are from the UK, Australia, and the US, with a few people from Europe. So it is global in the sense that it's happening worldwide, but it is only English speaking folks. So also kind of keep that in mind. Majority male, majority heterosexual, but a good chunk of sexual minority folks as well. And they asked people not only about their use of kind of the traditional party drugs, but also their use of alcohol, marijuana, and a large variety of other drugs as well. They asked about people's use of drugs, not just in these kind of party situations, but also in their loving, monogamous, consensual relationships, any kind of sexual situation. And then finally, something I really loved about this study is that they asked 10 different questions about why people are using drugs, and that included not just reducing shame, but also a variety of positive aspects, like are you using it to increase your desire, your arousal, to make yourself feel more sensation, to feel more connected to your partner, and so on. So let's take a look at what they found. I'm going to be focusing just on talking about the findings in women because that's the one that's kind of most pertinent to my pilot project and my research. So first and foremost, they asked people, when you use drugs or alcohol, how often are you doing so with the specific intent of enhancing your sexual experience, right? Not just that it happens to happen at the same time, or that, that there's some sort of coincidence, but that this is your specific intent. This is why you are using these drugs or why you are using alcohol. And so they asked people, and it's actually kind of, it is kind of amazing how many people said that they use drugs less than half the time, about half the time, more than half the time, or even always. And you can see even just in this breakdown that the sexual minority women, the lesbian and bisexual women, are reporting higher use of drugs specifically to enhance their sexual experience relative to the exclusively heterosexual women. And if we lump together all of the categories of people who say that they use drugs at least some of the time to enhance their sexual experiences, we see that in terms of the sexual minority women, we see that over a third of the bisexual women say that they do this at least sometimes, which is pretty remarkable. And it's kind of an understudied phenomenon to try to understand this. So if we then turn to looking at the different kinds of drugs that people are using, not surprisingly, people are reporting using alcohol and cannabis most often for their sex-linked drug use. Not surprisingly, because these are the drugs that are most commonly reported to use overall. I think caffeine is more slightly used than those two, but only by just. But again, even if we start looking at the individual types of drugs or alcohol, we see that the sexual minority women, in particular the bisexual women, are reporting using these drugs in connection with sex more often. Interestingly, the lesbian women are reporting either similar rates or much lower rates than the heterosexual women. So this suggests that it's not just being a sexual minority, but there's something specific about bisexuality that seems to be driving this difference. In terms of why people are using these drugs, I'm just going to pull out a couple of the interesting differences. So they asked women, which drug, if you were trying to increase your physical arousal, or which drug do you use to try to increase your physical sexual arousal? And so across the board, women are reporting that alcohol is the drug that they tend to use to increase their physical arousal. Both the hetero and the lesbian women identify this, and the bisexual women identify this as well, but they reported 4 times the alcohol use in order to increase their physical arousal than did the hetero or the lesbian women. In terms of increasing their sexual desire, again, alcohol comes out on top, with hetero women and bisexual women reporting twice the use of lesbian women, again, specifically to increase their sexual desire. And they asked a wide variety of questions around orgasm, which is really exciting to me because my research is in orgasm physiology. So they asked people, which drug do you use in order to have multiple orgasm? And the hetero and lesbian women are reporting cannabis primarily, and bisexual women also reporting cannabis as their primary choice, but reporting twice the level of the hetero and the lesbian women. So really interesting differences, bisexual orientation, and even by looking at different rationales about why people are using these drugs or alcohol in terms of enhancing which aspect of their sexual experience. So why do we care about all of this? Well, it's a huge public health issue because when people are intoxicated, it's a lot more difficult for them to give consent, a lot more difficult to negotiate consent in an ongoing fashion across the sexual experience. There's been a lot of research on this in alcohol and to a lesser extent in cannabis. In addition, there's problems with impaired contraception. So not only are women at a disadvantage in negotiating use of condoms or other barrier protection during their sexual activity, but ingesting alcohol or using cannabis can actually interfere with hormonal forms of birth control as well. So it can actually directly impair contraception as well. And then for drugs that are smoked, there's actually some really interesting research that suggests that smoking prior to engaging in oral sex really strongly increases the risk of developing oral STIs because it reduces mucosal immunity. So there's all these negative effects, side effects of using drugs in the context of sexual activity. But there's also this possibility that this may constitute a pathway by which drug use can become more heavy over time, or that it can lead to more problematic patterns of drug use. And that's kind of one of the things that I'm really interested in my RDAR pilot project, is looking at young women, college-age women who are using alcohol and marijuana in these sexual contexts. And does that then lead them to higher drug use over time, more problematic drug use patterns over time? Now, I really loved this study because it kind of started to answer some of these big questions and start to fill in some of the gaps in the literature. But one giant frustration that I have with this study that I just talked about, and pretty much all of the research on sexual orientation and drug use, is that it leaves out a really important sexual minority group, which is the heteroflexible or mostly heterosexual group. This is by far the largest sexual minority group in women. There's been a number of studies now looking at college-age women, and finds that 15 to 20% of college-age women identify as mostly heterosexual when they're actually given that option on a survey. And those numbers are increasing. Actually, at UNL in this last year, when we did a large-scale survey of the undergraduate population, the numbers were closer to 25 to 30% of college-age women were identifying with this label. So, you know, it's a very large sexual minority group. And yet, traditionally, sex researchers have lumped this group together either with the exclusively heterosexual women or with the bisexual women, despite the fact that there's really good evidence that this group is qualitatively different from both of those other groups. And one way in which they're different is in their drug use. So for example, they are 3.5 to 4 times more likely to report using alcohol and or marijuana than exclusively heterosexual women. And the few studies that we have looking at sex-linked drug use in heteroflexible women suggest that they're even more likely than bisexual women to report using alcohol and marijuana before sex. So this is a particularly vulnerable group about which we know particularly little. So my RDAR pilot project is going to be looking at heteroflexible or mostly heterosexual women. So it's the heteroflexibility, arousal, and substance use habits HASH study. Thank you, RDAR, for letting me use that acronym. And so in this study, we're going to be looking at 2 different possible explanations or 2 different pathways by which mostly heterosexual women may come to have this greater use of alcohol and or marijuana for sexual enhancement through looking at the reward value associated with sexual stimulation and looking at their sexual arousability, their physiologic sexual response. So starting first with looking at this reward value. So in terms of reward value refers to how motivated somebody is to seek out and engage with a particular stimulus. So for example, there are people in the world, I am told, who don't like chocolate, right? Like, I am a huge chocoholic. I will go to the ends of the world for good chocolate. And if you give me a piece of chocolate and you give a piece of chocolate to somebody who's just not all that into chocolate, and you were to look at the way that our brain processes that reward, the person who's kind of like meh about chocolate, their brain will respond to it, right? It does still have fat and sugar in it, right? But my brain is gonna, my mesolimbic reward system is gonna light up like a Christmas tree, whereas theirs might just kind of be like, okay, this is cool, right? And that's an individual level difference. I'm sorry, Ken, I know that was a horrible oversimplification, but so there are individual differences in the level of reward and reward activation that we have for different kinds of stimuli. So behaviorally, one of the ways that has been used to measure reward value is with the key press task. So in the key press task, you show pictures of something that is rewarding. So this has been done with pictures of attractive people, pictures of babies, pictures of food. And you present this image for a set amount of time, a second and a half. And if the participant presses the up key, then it will increase the amount of viewing time. And if they press the down key, it will decrease the amount of viewing time. This is essentially a lever pressing task, the same way that we do with rats, just with humans. And what we're measuring is how many times does this person press the up key or the down key. And this reflects their willingness to engage with that stimulus, to do some small amount of work in order to continue to engage with that stimulus. And in other circumstances, like in pictures of food, this has been shown to correlate really tightly with people's reward processing in response to that image. What shocks me is that this has never been used in sexual situations for sexual stimuli. And so one of the first things that we're going to be doing in this project is developing this key press task to look at sexual reward value. So we're going to be showing women sexual pictures. We're going to be doing this with exclusively hetero, mostly hetero, and bisexual women. And we're going to be looking at two different aspects of their sexual reward value. So first, we're going to be looking at their response at different intensity levels, showing them either pictures of hardcore pornography or softcore pornography. This is as close as I could come to a socially acceptable picture to represent that. We're also going to be looking at response to a diverse range of stimuli. So what are women's responses and their level of engagement with pretty vanilla pornography, more kinky pornography, pornography featuring different forms of different body types, a diverse range of actors and actresses, and then also comparing their responses to pictures of food. And so we're going to be looking at what are women's motivation, what is their reward value associated with these different kinds of stimuli characteristics, with the hypothesis that the mostly heterosexual women are going to show higher reward value for sexual stimuli, and that this will be reflected by their motivation, that is their number of key presses at lower levels of intensity and to a wider range of stimuli. So essentially, if we find that women, that mostly heterosexual women have a higher reward value associated with a wider range of sexual stimuli, then this would indicate that they are going to be more motivated to seek out sexual reward, including potentially by enhancing it by using drugs or alcohol. But that's only one piece of the picture, right? Because your initial approach and the reward that you get from a sexual stimulus, a visual sexual stimulus in particular, is only going to carry you so far, because then there has to be a physiologic response to that stimulation, right? If you engage with a stimulus, but then your body kind of goes, meh, it's okay, you're not going to be motivated in the future. That behavior is not going to be conditioned in the future. What we really want to see is that in combination with women's arousability, the degree to which they are able to have a physiologic sexual response to a range of sexual stimuli. So how do we quantify women's sexual response? Well, we do this with vaginal photopathismography. The vaginal photopathismograph is a device that we use to measure vaginal blood flow in response to sexual stimuli. It's this little device here. The science writer Mary Roach called it Cinderella's tampon. So this device has a photovoltaic cell and a light-emitting diode housed within an acrylic tampon-shaped device. Women self-insert this in the privacy of a locked room, and then we show them a series of different pornographic videos and varying intensities as well. And we can actually look at their second-by-second changes in genital blood flow. So we can look at both how quickly their body responds to a particular stimulus, as well as the total response over the course of the video. So we can look at their arousability as well as their total arousal level. So looking at these two different pathways, we're going to be looking at, is there a difference in how mostly heterosexual women's sexual reward and their arousability interact in ways that predict their self-reported use of drugs for sexual enhancement. And in particular, we're going to be looking at this in combination with or above and beyond what would be predicted from more traditional models of sexual minority disparities in drug use. So looking at things like minority stress, sexual trauma history, and mental health. So do these sexual responsivity variables predict people's use of drugs for sexual enhancement above and beyond the known variables, the known models of why sexual minorities might use drugs at a higher rate? So that's the project that we're going to be running just as soon as we are allowed to do in-person research again. And then kind of future directions, which I'm really excited about, we just submitted a supplement to the RDAR award to look at these same pathways, but to also include consideration of nicotine use. So there's been some recent studies that have come out that suggest that sexual minority women, and in particular bisexual women or bisexually oriented, which includes the mostly heterosexual women, that they are not only more likely to use nicotine, but they're more likely to use nicotine in conjunction with alcohol and other drugs, and they're more likely to use nicotine during or prior to sexual experiences. So that's really interesting because nicotine can be an amplifier for reward. So the interaction of the fact that these women might be more likely to be using nicotine in a sexual situation while they're also using drugs in a sexual situation might interact with these other variables in ways that even further perpetuates this differentiation in terms of what they're then conditioned to do to seek those rewards again in the future. So that's kind of, fingers crossed that we get that supplement so that we can go even further into this model, but I will go ahead and stop my screen here.
So it's my great pleasure to introduce our last speaker, who's from our fellow Cobra on the opioids and overdose, Jackie White Hughto. She's an assistant professor at Brown University, and she's from the Department of Behavioral and Social Sciences. And she's also an assistant professor at the Department of Epidemiology. Her project title as project leader is Combat. I have to come up with these great acronyms. Combat. I got nothing. Combat Opioid Use Disorder, a pilot RCT of combined medication and behavioral activation treatment for people living with opioid use disorder. Jackie, please take it.
Great. So today I'll be starting with an article that's relevant to my topic and relevant to my Cobra funded project. Thank you for giving me the opportunity to share this work with you all today. So my project is, as Rick mentioned, is combat combined medication and behavioral activation treatment for individuals living with opioid use disorder. It specifically focuses on helping people who are receiving medications for opioid use disorder stay engaged in care and reduce or maintain their reductions in substance use. So when I was thinking about articles to present, at first I thought about, do I present behavior activation? But I know kind of the focus was to present something interesting and novel, and certainly one of the things that I'm currently dealing with as a researcher and also as someone who's looking to support individuals who are in treatment is how COVID-19 has affected care. So the article is called Opioid Use Disorder and COVID-19, Crashing of the Crisis. And this was a commentary by two treatment providers. And it was written in late March of the epidemic. So, kind of right at the beginning as care centers kind of transitioned into providing remote care delivery. And so some of the things that happened immediately following the social distancing guidelines were that jails and prisons started to decarcerate in order to mitigate COVID-19 outbreaks within these facilities. We also saw that because of social distancing guidelines, that presented challenges for social isolation. And we know that individuals who are using opioids in particular alone are at greater risk for fatal overdose. Similarly, during this time, of course, we're all experiencing greater kind of mental health stressors with the anxiety of the outbreak and the economy. And so this kind of, these stressors certainly can be a trigger for substance use disorder uptakes and use, as well as relapse. And so in terms of changes that have happened that have actually improved care delivery, on March 17th, 2020, the DEA reduced its restrictions on buprenorphine initiation, which allowed individuals to access the drugs via telehealth. So overcoming a huge barrier that folks have been working on for a long time in order to increase accessibility to buprenorphine as a treatment for opioid use disorder. We also saw restrictions lifted in methadone take-home. So individuals, there used to be restrictions on how many methadone take-homes you could have at a time because of concerns around diversion. And so those restrictions were lessened, allowing people to not have to go into the clinic for daily dosing in many cases. We also saw expected, there's kind of a discussion around expected changes in the supply chain because of transportation or shipping changes and kind of like the flow of drugs internationally, that would expect to see, less of lessened availability of street drugs, which could increase the price, potentially leading more people into treatment when to manage their withdrawal symptoms. And so all of these kind of changes as a result of COVID-19 have potentially produced innovations within the state of care. This article listed a number of current and recommended innovations for care delivery. So some of the things that have been happening in clinics involve the use of peer recovery specialists who are using texting and video conferencing for outreach in order to maintain strong connections to patients. We also see case management and social work services being provided by phone and video chat in many cases. Buprenorphine prescribers are recommended to, and in actuality are providing longer durations of prescriptions so that, again, people don't have to come into the clinic as frequently. And all of these kind of regulations have allowed for the immediate deployment of telehealth. And we know that telehealth has worked for a variety of clinical care, both in terms of medication prescribing, but also in terms of counseling. And so people can access telehealth via their computer or phone. But certainly, while telehealth can increase access for a number of people, it can also create inequity challenges for a number of people. So individuals who don't have access to cell phones or have limited minutes, some of the innovations that clinics are doing are, providing access to cell phones, either like physically giving cell phones to people or having sanitized stations within the clinic where people can go and use phones to be able to have telemedicine appointments. Also pharmacies, this one thing that can certainly happen is they can serve as hubs for telemedicine, so people can go in and access their care in a pharmacy. And so I can save these for the end. And I know that everyone's kind of coming from different backgrounds, work on different kind of aspects of substance use and treatment. But I'm kind of wondering how much these points that were raised by the commentary ring true to all of you in terms of your patients or participants, or what you've read and learned about and observed during the COVID crisis. And if there's any specific challenges that are unique to people who use drugs that you've observed that were not captured here. And then also, like, what other kind of innovations have happened in terms of care delivery that really should be carried forward as we think about improving access to care for people living with opioid disorder and other substance use disorders. So again, we can round back to this at the end if that is helpful. But going into the topic of my particular presentation, so combat opioid use disorder, just to give you some background on the project, we know that medications for opioid disorder, including methadone, buprenorphine, and naltrexone, are effective in reducing opioid withdrawal, cravings, and fatal overdose. But one of the challenges is long-term retention and care for these treatments. We find that this is often suboptimal. And while there are many drivers of this, one kind of large underlying mechanism is depression. We find that 20 to 61% of people entering opioid treatment have major depression. And depression, of course, can be caused from a number of biological, behavioral, and psychosocial challenges. So both the biology of taking opioids can lead to depression, also life situations that result from chronic opioid use can lead to depression. And certainly this includes the stigma of having opioid use disorder and the stigma of treatment. And we know that people with depression and opioid use disorder are more likely to drop out of treatment and relapse than those who are not depressed. So it really makes sense to target depression as a means of helping people stay engaged in care. So this particular study uses behavior activation, which is evidence-based treatment for depression, and to posit that it could be useful in boosting the efficacy of medications for opioid use disorder. And BA posits that life events lead individuals to experiencing low level of positive reinforcement, thus leading them to engage in self-defeating behaviors like isolating, avoiding, and even using drugs to cope. And so these self-defeating behaviors may make individuals feel better in the short term, but ultimately they can increase and exacerbate depression through a cycle of negative reinforcement. So what BA does, so behavioral activation is basically the B in cognitive behavioral therapy for those who are familiar with CBT. And what it does is it's less intensive to deliver. It's easier to train counselors who don't have a PhD in psychology. And so it focuses on helping individuals increase goal-directed, rewarding, and pleasurable activities. And by actually getting out and getting activated and doing things that can increase mood, individuals experience less depression, and then they're able to ultimately, hopefully, engage in care and manage their substance use. And so BA has been used in a variety for treatment for a variety of different substance use disorders, including smoking, alcohol, and crystal methamphetamine use. And so this would be the first time that it's being tested explicitly in a community care setting with the goal of helping individuals stay on medications for opioid use disorder and reduce their substance use. And so the pilot project is ultimately, as part of this project, my colleagues and I adapted existing behavioral activation interventions for opioid use disorder, and we're evaluating the feasibility and acceptability of the intervention. And so just to give you a brief overview of who's involved, it's folks 18 years of age or older. They're prescribed methadone or buprenorphine, specifically Suboxone or Subutex, taken daily. And to be eligible, they had to have missed one dose of methadone or buprenorphine or a clinical visit in the past 30 days. They also have to have moderate to high depressive symptoms. And so as far as recruitment so far, we've primarily been working with substance use treatment clinics for direct referrals, as well as being on site at these clinics prior to COVID-19 and using passive recruitment strategies like flyers and things of that nature. We've also worked with the various community partners around Providence, Rhode Island for direct referrals. So just to give you a quick overview of the way the intervention and study design is structured, all folks come in for a baseline visit. Everybody gets 2 sessions of standard substance abuse treatment and health navigation sessions, including overdose prevention. And these are curricula that we developed drawing on SAMHSA and protocols that were used by some of our treatment partners. Then folks are randomized to the control treatment as usual or the combat intervention. Both groups would continue taking medications as prescribed by their provider. We're not actually providing them with the medications. So those in the control would just come back for 8 weeks and just do a weekly assessment and provide urine for toxicology screens. And those in the intervention would go on to get the 8 weekly BA sessions, as well as the brief survey and tox screen. And then everyone comes back at 3 months for a follow-up assessment, and again at 6 months. We're using a variety of data sources. We're primarily relying on qualitative and quantitative data collected at the study visits, but we're also working with state registries to get behavioral data and prescription drug data to be able to triangulate self-report of medication use and treatment engagement. And then working with our community partners, we'll be supplementing all these data with patient records. So all of this has been challenged. As Tierney mentioned, her in-person data collection is on hold. As soon as COVID-19 happened, we immediately switched to remote procedures for anyone who's currently enrolled. So people could complete their therapy sessions online with a counselor or by phone if they didn't have access to the phone. And now we're waiting IRB approval to begin remote enrollment. And so we're recognizing how this is really going to change our participant population in many ways. Many people don't have access to reliable internet, and the therapy isn't really designed and can't really be adequately delivered by phone. So we're going to start this initial remote enrollment. And then once we're able to be in person again, we'll offer people either remote or in person. And so we'll be kind of evaluating the feasibility and acceptability of these different procedures. But notably, as I mentioned with my article early on, and it's true of the providers that we're working with locally, folks have really switched, all the clinics have switched to providing remote counseling. And so patients are very familiar with the use of remote counseling. And in many ways, it creates, increased accessibility for people who don't have to travel to the clinic. But of course, folks who don't have access to these forms of technology, it may actually widen some of the inequities. But this is a feasibility and acceptability project. Our goal is to get those 80 folks enrolled, hopefully by March 2020, evaluate the data, and then if everything is trending in the right direction, we'll write an R01 to do a larger RCT of the intervention. So that is what I have.